digestion of a protein, we have mainly two sources. So one is exogenous source. So exogenous source means that which we generally include on our food. So and this protein may be from animal source or from vegetable source. So around 50 to 100 gram of protein we generally daily we consume. And apart from that, this for the digestion of a protein, we get these endogenous sources also. That means these enzymes, the enzymes which is present in a digestible juice, and these epithelial cells of our intestine. So this becomes a good source for digestion of a protein, and it contributes around more than this one daily consumed protein. And the enzymes, so generally we consume a large amount of proteins, but only a very few amounts of nitrogen is excreted in a fish daily. So that means most the large amount of proteins what we consume, they are digested and they are easily absorbed in our body. So very few only it is excreted. So this protein digestion has a greater efficiency. So we have a different sites where the enzymes for a protein digestion is present, and this so it is the enzymes that pepsin. So it is present in the stomach, and other enzymes that is from a different source. They are the digestion takes place in the intestinal lumen. So it is pancreatic enzymes, so which is present in a pancreatic juice. So the enzymes like this trypsin, chymotrypsin, carboxypeptidase, elastase, and similarly the enzymes present in the intestinal juice, which is responsible for digestion of proteins, are in enteropeptidase, aminopeptidase, dipeptidase, and even in a mucosal cells like this dipeptidase and trypeptidase. And these all enzymes which is responsible for a digestion of a protein, they are generally called as a proteolytic enzyme. So these enzymes are called as a proteolytic enzyme. And these proteolytic enzymes, they are grouped under a class hydrolase, and basically they can be divided into the two groups. So one is endopeptidase, the proteolytic enzymes which cleaves the internal peptide bond. So they are called as endopeptidase. And next group, they are called as exopeptidase, so which cleave of one amino acid at a time from either end of a molecule. So those enzymes which remove one amino acid from an N terminal of a polypeptide, these are called as aminopeptidase. And those enzymes which remove one amino acid from a C terminal of the peptide called as a carboxypeptidase. So these proteolytic enzymes, so they can be grouped according to the site of actions over a peptide bond. So they can be divided into two groups, endopeptidase and exopeptidase. So endopeptidase enzymes, so they cleave the internal peptide bond. And exopeptidase, so they remove a amino acid, single amino acid from a either from a N terminal. So those enzymes which remove amino acid from a N terminal called as a aminopeptidase, and those remove amino acid from a C terminal of a peptide bond known as a carboxypeptidase. And one important thing regarding about these proteolytic enzymes, so all these proteolytic enzymes, they are secreted in the form of zymogen. So the zymogen means that is the precursor form. So during a secretions, they are not in an active form. When they get a contact with its substrate, then only these enzymes, three enzymes, they are converted into the active enzyme. So these enzymes which is responsible for catalyzing protein or digestions protein, they are called as the proteolytic enzymes. These proteolytic enzymes are two types, endopeptidase, exopeptidase, and all these enzymes, they are secreted in a lumen in the form of precursors form. So that is a zymogen. So inactive form, zymogen means that is an inactive form of enzyme. So this is a figure. So endopeptidase, they cleaves the inner side of peptide bond. Exopeptidase, they remove a single amino acid from a each terminal. So aminopeptidase from N terminal. So dipeptidase from the two amino acids. This dipeptide at a time from a N terminal. Tripeptidase is remember 3 amino acids. So tripeptide from N terminal. So carboxypeptidase they remove a single amino acid from a C terminal. So peptide dipeptidase it's remove 2 dipeptide from a C terminal, and this dipeptidase it breaks the dipeptide into the 2 free amino acids, and this tripeptidase so it's remove a single it acts on a tripeptides and remove a single amino acid from a N terminal. So this is the example. So depending on the source of these peptidase or these proteolytic enzymes interact with a substrate in a digestive lumen, so they divided into 3 stages. So one is gastric digestions, next one is the pancreatic digestions, and next one is the intestinal digestions of a protein. So since these proteolytic enzymes, so they are not present in a do not take place in the mouth. So the digestions, unlike these digestions of carbohydrate, the digestions of protein, it is start from a stomach. So in previous class we studied the digestions of protein. They do not, the digestions do not takes place in the mouth due to the lack of the enzymes, and this it is start from a stomach. So when this food in containing these proteins they reach to the stomach, then it stimulates gastrin, and this gastrin in turn it stimulates the secretions of the releasing of gastric juice, so which contains hydrochloric acid and proteolytic enzymes, majorly pepsin in case of infant, and this renin is also there, but it is not important in a adult, and this renin is important in case of the infant, where this staple diet is this one so this hydrochloric acids, which is present in the gastric juice, it is secreted by cells. And the main functions of these hydrochloric acids have a strong bacteriostatic actions, as well as it also help to denature the protein. And apart from that, it is also necessary for the activations of pepsin. So initially, this pepsin, so which is the this active enzyme, it is secreted as a pepsinogen. So pepsinogen is the inactive form of pepsin. In simply we say that this pepsinogen is a zymogen. So these conversions of pepsinogen to the pepsin, it require HCl. Apart from that, is hydrochloric acid. So it's an acid, so being it that it creates a acidic environment, and this acidic environment is favorable for the action of a pepsin. So this pepsin, so it is secreted by a chief cell as a pro enzymes or as a zymogen, as a pepsinogen, and this pepsinogen is converted into the active pepsin first by HCl, and after the formations of this pepsin, then this pepsin is responsible for a conversions of other pepsinogen to the active pepsin, so which is called as a autocatalysis. So pepsinogen is converted to the active form of pepsin with in presence of HCl and pepsin itself. So by removing a 42 amino acids from a N terminal to form a active pepsin, and this pepsin is a endopeptidase. So pepsin enzyme, so it is a endopeptidase. It is an endopeptidase, and it is active at pH between 1.6 to 2.5. So when the pH is increased more than 5, then this pepsin gets inactivated. So this endopeptidase means that acts on the peptide bond of the internal peptide bond. So it acts on the internal peptide bond. So though it randomly acts on the internal peptide bonds to cleave this protein into a different peptides, so the site of this the activity that the action this activity of pepsins they have a specificity, so they can only cleave on a specific site. So those peptide bond which carboxyl groups, which carboxyl group is contributed by a aromatic amino acids, so such as this phenylalanine and tyrosine, and acidic amino acids, so that is a aspartate and glutamate. So generally we should know about that is peptide bond. So generally this peptide bond is formed by a amino group and carboxy group. So during a formation of a peptide bond, so one amino acids it contributed its amino group, and the next amino acids contributed carboxy group. So this carboxy and amino group, so it's from a peptide bond, and this pepsin is able to cleave those peptide bonds, so which carboxy group is contributed by specific amino acids such as aromatic amino acid like this phenylalanine and tyrosine, and acidic amino acid like this glutamate, that's a glutamic acid, and aspartate, so that is a aspartic acid. So glutamate, aspartate, and phenylalanine and tyrosine, so which carboxy group is contributed by aromatic amino acids and acidic amino acid. So after the actions of this pepsin over a protein, it degraded this protein into the large peptides and some proteases and some peptones. So these protein molecules, by the actions of pepsin, is converted to the these different peptides. So renin, this renin is also present in a gastric juice, but it is more important in case of the infant. And this renin is like pepsin; it is also secreted as a prorenin, zymogen, and its activities. This renin is more active at pH around 4. And generally, this renin is acts on a casein of a milk and is involved in a curdling of a milk. So the substrate for activity of a renin is a milk, and however, this renin enzymes that is less inactive in case of adults, since the pH of stomach is below 2. That is highly acidic, so on which this renin gets inactive. And in case of this one infants, due to the stable diet they consume milk, so that which maintain this pH of a stomach is around slightly, that is slightly acidic, so not highly acidic. So where this renin is more active, and they also it get this good substrate to catalyze. So now this next phase, that is the pancreatic phase of a digestion of protein. So as soon as these peptides, this partial digested of a proteins, so by pepsin in a stomach, that pepsin cleaves protein into the peptides. So along with food, these peptides they reached into the small intestine, and when these peptides they reach into the this intestine, it triggers the secretions of hormones such as this cholecystokinin and secretin, and this cholecystokinin and secretin hormones, so in turns they stimulates the pancreas to release a pancreatic juice, and this pancreatic juice contains a bicarbonate, so which is responsible for maintaining alkaline environment over intestinal lumen, and it also contains different proteolytic enzymes like trypsin, chymotrypsin, elastase, collagenase, and carboxypeptidase. So this trypsin, chymotrypsin, elastase, collagenase, and carboxypeptidase. So these are the proteolytic enzymes which is present in a pancreatic juice. So trypsin, so this trypsin is secreted like this pepsin. This trypsin is also secreted as a zymogen, that is a trypsinogen, and it is activated. That trypsinogen is converted to the trypsin in presence of a enterokinase enzymes. And this enterokinase, so this is the enzyme. It is secreted by a intestinal mucosa. And in the initially enterokinase, it converts trypsinogen to the trypsin, and once the trypsin is converted, then same trypsin is able to convert trypsinogen to the further active. So the conversions of trypsinogen to the trypsin by removing six amino acids, so as a hexapeptide. So like this one, it's like pepsin. It is also a endopeptidase, so which cleaves a internal peptide bonds presence in a peptides, and it specifically catalyzes those. It hydrolyzes those peptide bonds which carboxy group is contributed by a basic amino acid. So those peptide bonds which carboxy group is contributed by a basic amino acid, such peptide bond is cleaved by a trypsin. So these basic amino acids, so like lysine and arginine. So those peptide bonds which carboxy group is contributed by lysine and arginine, such peptide bonds are cleaved by trypsin. So it is an endopeptidase. And apart from this hydrolyzing of peptides, which carboxy group is contributed by a basic amino acid, this trypsin is also necessary for the activations of other pancreatic proteolytic enzymes such as this chymotrypsin, elastase. So this like this pepsin and this trypsin, this chymotrypsin and elastase, they are also secreted as a zymogen form. So that chymotrypsinogen and proelastase. So the conversions of chymotrypsinogen to the chymotrypsin and proelastase to the elastase. So for that, it require a trypsin. So trypsin converts this inactive form to the active form. Apart from that, this trypsin is also required for conversions of fibrinogen to fibrin. However, it has a weak action on a casein. So trypsin inhibitors are also present in some foods. So like the plant's food, like the soybeans, peas, beans, wheat, and in a pancreas or cholesterol, so which contains a trypsin inhibitors, and this prevents the action of a trypsin. So chymotrypsin. So chymotrypsin is secreted as a chymotrypsinogen from a pancreatic juice, and the conversions of this chymotrypsinogen to the chymotrypsin is done by a trypsin. And this like this pepsin and trypsin, this chymotrypsin is also a endopeptidase, and it cleaves the internal peptide bonds present in a these polypeptides, which carboxy group is contributed by a aromatic amino acid like this phenylalanine and tyrosine. And apart from that, those peptide bonds which carboxy group is contributed by a leucine and methionine. So those peptide bond, so it cleaves a polypeptides, uh, which peptide bond is contributed by this carboxy group is contributed by phenylalanine, tyrosine, leucine, and methionine. So elastase. So elastase is also secreted from a pancreas juice in the form of proelastase, and in presence of trypsin, this proelastase is converted to the elastase, and this elastase, it is also endopeptidase, and it cleaves a peptide bonds which carboxy group is contributed by a these small amino acids, so like alanine, serine, and glycine. So elastase cleaves a peptide bond which carboxy group is contributed by a small amino acid like alanine, serine, and glycine. And collagenase, so it acts on a collagen. So carboxypeptidase, so these carboxypeptidases are the exopeptidase, so which is secreted by a pancreas. So it is also synthesized as a zymogen, that's a procarboxypeptidase. and this conversions of this pro-carboxypeptidase to the carboxypeptidase. For that also it requires a trypsin. So trypsin is responsible for conversions of pro-carboxypeptidase to the carboxypeptidase. And this carboxypeptidase of two types. So carboxypeptidase A, carboxypeptidase B, and this carboxypeptidase is a exopeptidase. So it's remove one amino acids at a time from a C terminal of a polypeptide. So carboxypeptidase A. So it's a metalloenzymes which contain a zinc, and so the activations of that conversions of pro-carboxypeptidase A to the carboxypeptidase A is done by a trypsin. So trypsin is necessary to convert this active form of carboxypeptidase A, and this carboxypeptidase A, it's remove hydrophobic amino acids. So when this on C terminal, if there is the presence of hydrophobic amino acids, then this carboxypeptidase A removes these hydrophobic amino acids at a time. So hydrophobic amino acids. So it's glycine, alanine, leucine, isoleucine, valine, proline, methionine, phenylalanine, and tryptophan. So uh, this carboxypeptidase A acts on the those C terminal, which on which the C terminal contains these hydrophobic amino acids. So carboxypeptidase B. So it is also a metalloenzymes, and it also this pro-carboxypeptidase B converted to the carboxypeptidase B. So the activations, for its activations, it also requires trypsin, and it releases the basic amino acids. So it acts especially on those peptides which carboxy terminal is formed by a basic amino acid like this lysine and arginine. So it releases arginine and lysine from a C terminal of a peptide. In summary of these digestions on these intestinal pancreatic juice and the stomach, so that's the pepsin, which acts specifically on those peptide bonds which carboxy group is contributed by aromatic amino acids and acidic amino acid. Trypsin, it is also endopeptidase. It cleaves peptide bonds which carboxy group is contributed by lysine and arginine. So the basic amino acids. Chymotrypsin, it is also endopeptidase which cleaves a peptide bond which carboxy group is contributed by aromatic amino acid. Elastase, so it is also in the peptidase which cleaves a peptide bond which carboxy group is contributed by these small amino acids like this alanine, serine, and glycine. Carboxypeptidase A, so it's remove a single amino acid from a C terminal of polypeptide. So if the C terminal of that polypeptide is hydrophobic amino acid, then it's remove these hydrophobic amino acids from that polypeptide by the actions of carboxypeptidase A. So it's a exopeptidase, and it's carboxypeptidase B. So it's also a exopeptidase, so which removes these basic amino acids like lysine and arginine from a C terminal. So now there is the intestinal phase of digestion of protein. So it also takes place in the intestinal lumen, but it is this digestions, for the digestions, these proteolytic enzymes, they are secreted by a epithelial cell in the form of intestinal juice. And this intestinal juice, it contains a different proteolytic enzymes like this enterokinase, aminopeptidase, prolidase, and di and tripeptidase. And these aminopeptidase, prolidase, di, and tripeptidase, these all are the exopeptidase. So this is the intestinal juice. It generally releases a proteolytic enzymes which are exopeptidase in nature. So enterokinase, so this enterokinase, it is released from an intestinal juice, and it is majorly necessary for the activation of a trypsin. And generally, this enterokinase is present in the epithelial cells of a brush border of a duodenal mucosa, and bile salts help in its liberation into intestinal lumen. So aminopeptidase, so these are the exopeptidase. It hydrolyzes the peptides bond next to the N-terminal amino acid of a short peptide. So it releases the single amino acid at a time from a N-terminal. For this, it's require a or manganese ions as a coenzymes for their activity. So these are different types of aminopeptides are there. For example, this leucine aminopeptidase, so which remove a leucine from a N-terminal. So next, these are prolidase. So it's also exopeptidase, so which remove a proline residue from a N-terminal end of a peptide chain. Prolidase, leucine, peptidase, it's remove a leucine from a N-terminal of a polypeptide, and it's prolidase, it's remove a proline from a N-terminal of a peptide chain. So di and tripeptides, peptidase. So digestions of this di and tripeptides is brought about by a di and tripeptidase present in the breast border membrane of epithelial cells as well as present in the interior of the cells. These di and tripeptidase, they are not only present in the intestinal lumen, it is also present in inside the epithelial cell also. So these di and tripeptidase, so it cleaves a substrate, dipeptides and tripeptides. So this dipeptidase, it converts dipeptides to the free amino acids, and tripeptidase, it converts tripeptide to a free amino acids and a dipeptide. And then again, this dipeptidase acts on a dipeptide to the free amino acid. And this dipeptidase, it's require a cobalt or manganese ion for their activity. So this is associated with the digestion of proteins. So acute pancreatitis. This acute pancreatitis is a premature activations of pancreatic proteolytic enzymes in the pancreas itself. So that causes a digestion of secretory mucosa, causing acute pancreatitis. It's a life-threatening condition. So generally, what happens is all the cells, not only the digestive cells, other cells, that's the pancreas cells also, they are also composed of proteins. So when these enzymes, these proteolytic enzymes, if they activated in this pancreas itself, then what happened? These enzymes now started the digestions of pancreatic cells, so which destroy pancreatic cells, so causing a acute pancreatitis. And this is the one of the reasons this all these proteolytic enzymes, they are secreted in a zymogen form. So that is the inactive form. So if these enzymes, they are secreted in the active forms, then what will happen? So simply they start to digest the own cells. So to prevent the digestion of that own cell, these enzymes, proteolytic enzymes, are secreted in zymogen form or inactive form. So this is associated with the digestions of protein. So we are discussing on. So the most common disease that associated with the digestions of proteins, that is the acute pancreatitis. So generally these proteolytic enzymes, they are secreted in the form of zymogens. So sometimes what happens, these proteolytic enzymes, they get activate in their host cells. That means that is in the pancreas itself. So when these enzymes, they get activated in a pancreas, then they start to digest the pancreas cells itself. So which cause a acute pancreatitis. It's a serious disorder. So next disorder, that is the steatorrhea. So steatorrhea, so in generally in conditions of a deficient pancreatic secretions like cystic fibrosis, chronic pancreatitis, or surgical removal of a pancreas, the digestions and absorptions of fats and proteins is left incomplete. So that these proteins along with the fat, they are accepted in a feces. So if there is the more amount of these fats and proteins, they are present in a feces, then the condition is known as a steatorrhea. So that's the large excretions of these proteins along with the fat. So generally, steatorrhea is a term used where there's a more fat is present in a feces. So there's a fatty stool is called as a steatorrhea. So during that time when these pancreas, they are not able to release these digestive enzymes, then there is the partial digestions of proteins takes place, so that they cannot get absorbed, and so they are excreted in a, so which is called as a steatorrhea. So now there is the absorptions of amino acids. So proteins are completely digested to the constituent amino acids. So like this carbohydrate, these proteins also, they are completely digested with the help of a different proteolytic enzymes present in this gastric and intestinal lumen. So these enzymes, they converted this protein into the free amino acids. So these proteins, they are absorbed in the free amino acid form. However, these peptides, they are remain undigested, and these peptides, they can also absorb from a intestinal lumen to the epithelial cell. So in case of this one, this absence of a carbohydrate, in case of carbohydrate, what happened? Only monosaccharides, they are able to absorb to the epithelial cell. But in case of these proteins, most of the protein, they are absorbed in the form of free amino acids to the epithelial cell. But some undigested peptides, they also able to absorb to the epithelial cell. And so these undigested peptides, so which we call as a oligopeptides, they are absorbed from a duodenum and proximal jejunum, whereas these free amino acids are absorbed from a distal jejunum and a ileum. So peptide absorption, so the absorption of peptide, it is also a active process, and for that it require a hydrogen. So these peptides, they co-transports along with the hydrogen ion, and this is a more active, more rapid than the absorption of a free amino acid. And after it enters to these peptides, they are entered to the epithelial cell. Then on epithelial cell, these peptides get degraded into the free amino acids with the help of the enzymes that is present in the epithelial cell. So already we have mentioned that this dipeptidase, tripeptidase, they are not only present in a lumen of small intestine, they are also present in the epithelial cell. So those peptides which is absorbed from the apical site of a intestinal lumen to the epithelial cell, these peptides are degraded into the free amino acids with the help of this dipeptidase and tripeptidase. So only free amino acids, they are able to absorb to the basomembrane side. So that is towards the blood from a epithelial cell. So generally these epithelial cells have two sides. One is the apical site, and another one is the basal side. So from apical side, here these peptides or amino acids, they are enters to the epithelial cells from a intestinal lumen, and on the basomembrane side, these amino acids, they are released to the blood from a epithelial cell. So free amino acid absorptions. So it is also a active transport mechanism, and the mechanism of these absorptions of this free amino acid is similar to the absorptions of these monosaccharides like this glucose and galactose during a digestion of carbohydrate. So why? Because for the absorptions of this glucose and galactose, it requires sodium. So it's here a sodium. It's required along with the sodium. These glucose and galactose get absorbed from intestinal lumen to the epithelial cell, and like that, these amino acids, for the absorptions of amino acids from a intestinal lumen to the epithelial cell, it also requires sodium. So the mechanism is same. So that this sodium transport from intestinal lumen to the epithelial cell down the concentration gradient. So that means the sodium is higher, they are present in a higher concentration in the intestinal lumen than the epithelial cell. So this sodium moves from higher concentration to the lower concentrations, and the force then this and these amino acids like carbohydrate, these amino acids transport from lower concentration to the higher concentration, and the energy required for this transport of amino acids against a concentration gradient is provided by a sodium, which moves towards the concentration gradient. And here the energy is required indirectly. It's also a secondary active transport where this ATP is necessary for a movement of this sodium to the from epithelial cell to the intermembrane space. However, there are though these amino acids, they share a sodium, so they share a co-transport along with the sodium for the absorptions of this amino acid from intestinal lumen to the epithelial cell. But there are different type of carriers. There are different specific carriers are available for a different amino acids. So at least 5 to 6 carrier proteins have been identified. So one carrier protein, which is only specific for a neutral amino acids, and this carrier protein that is only specific for a basic amino acid and cysteine. And next one is responsible for a carrying of acidic amino acid. And next, that is responsible for a carry of amino acid and glycine. And next one, that is responsible for a transport of beta amino acids such as beta alanine. And few amino acids they can transport by this facilitated mechanism also. So neutral and hydrophobic amino acids and some basic amino acid they choose a facilitated transport system for the absorptions. And however, these D amino acids are transported by a passive diffusion, and this active transport system is generally followed by a L amino acid. For the absorptions of L amino acid, this active transport system is necessary, and these D amino acids are transported by a passive diffusion. So some these amino acids, which is absorbed from intestinal lumen to the epithelial cells, they are some of them are get metabolized in epithelial cell itself. So such, for examples, that the synthesis of glutathione. So this glutathione is synthesized by a 3 amino acid, that is glycine, cysteine, and glutamate. So these 3 amino acids they are responsible for a synthesis of a glutathione, and which is takes place itself in a epithelial cells, intestinal epithelial cells. And next one is that is the arginine, which is absorbed from intestinal lumen. These arginine are converted to the citrulline in intestinal epithelial cell, and this metabolism is necessary because it prevents the conversions of this arginine to urea in the liver. So generally, this amino acid arginine, which is absorbed from the intestinal lumen to the epithelial cells, so this is converted to the citrulline in the epithelial cell. After that, this citrulline is released in the blood, which is transported to the kidney. And kidney again, what happened? This citrulline is converted to the arginine, and in this way, what happened? Arginine is regenerated, and this step prevents the formations of urea from arginine in the liver. And next things, these glutamate and aspartate. So these are acetic amino acids. So these are get transaminated in a enterocyte itself to their respective oxaloacids. This glutamate is converted to the oxaloacetate, and this aspartate is converted to the glutamate is converted to the alpha ketoglutarate, and aspartate is converted to the this oxaloacetate in a enterocyte. So because of that, what happened? The concentrations of glutamate and aspartate is found very low in the blood. Along with the glucose, some amounts of glutamine they are metabolized for the productions of ATP in the epithelial cell itself. So this glutathione, so it is also involved in the transport of certain amino acids, especially this glutathione is necessary itself. It is important for the absorptions or transport of neutral amino acids, L-amino acids. So generally, this glutathione, so there's a translocation absorptions of amino acids with the help of glutathione. So this is morely active in case of certain sites of a body like this small intestine, kidney, seminal vesicles, epididymis, and a brain. And generally, this cycle is also called as gamma glutamyl cycle or Mestier cycle. And in here, what happened? This first amino acids, those amino acid which is to be transported, so that is bound with a glutathione, and along with the glutathione, this amino acid is translocated into the cell in the form of glutamyl amino acid. So glutathione bind with the free amino acids, and which is translocated with the help of the enzyme gamma glutamyl transpeptidase, and so that this amino acid bind with the glutathione to form a glutamyl amino acid. Now this glutamyl amino acid is get releases in the form of amino acid with the help of the enzyme gamma glutamyl cyclotransferase, and now this time this glutamyl is converted to the oxoproline. And from this oxoproline, this with the help of the enzymes 5-oxoprolinase is regenerated to the glutamate. And for that, it requires energy, and this energy is provided by a ATP. And this glutamate is again bind with a cysteine to form a glutamyl cysteine. And for that, the energy is required, which is provided by a ATP. And the reaction is catalyzed by the enzyme gamma glutamyl cysteine is catalyzed from a glutamate. The enzyme is regenerated. And the purpose of this mysterious cycle or gamma glutamyl cycle is that is the transport of free amino acid. So oxoprolinuria, so that's a pyroglutamic aciduria. So generally it's a common, and it is majorly due to the deficiency of enzyme 5-oxoprolinase. So due to the deficiency of 5-oxoprolinase, that oxoproline, so that cannot convert it to the glutamate, so that it is accelerated in a urine. So absorptions of intact protein. The absorption of intact protein has a less into the nutritional value, so it's a less important. However, if we think about this immunological value, it has a great importance. And generally, these intact proteins are absorbed by means of a endocytosis or a pinocytosis, and so it is necessary. It's a very important in case of a newly born infants. So there is a direct absorption of intact proteins. So where is immunoglobulin A from a colostrum. So this colostrum, so which contains immunoglobulin A, and this immunoglobulin A is absorbed by infants from they get this IgA from a maternal this from its mother, and these intact proteins and polypeptides are not absorbed by the adult intestine. So if some these proteins they are absorbed in a adult, then it causes a food allergies. So like this gluten-induced enterofatty, fish allergies, or egg allergies. So in case of the infants, this absorption of intact protein is very important. This is the main way of a transport of this IgA from mother to the infant. So then this IgA is present in the colostrum, and if some these intact proteins, if it is absorbed in an adult intestine, then it's lead to a different undesirable immunological response. So that is a food allergies. So example of these food allergies are this gluten-induced enterofatty, fish allergies, or egg allergies. So next, this one is cystinuria. So defect on a dibasic amino acid transporter proteins result in a loss of these amino acids in a feces and urine. So unable to absorb of these dibasic amino acids. So that is the cysteine, ornithine, arginine, and lysine. So COAL. The next one, Hartnup disease. So this is the inability of absorption of tryptophan. So unable to absorb the neutral amino acids, and on that also this tryptophan absorption is severely affected. So that resulting to the pellagra. So this tryptophan is a source, so major source in our body for the synthesis of a niacin vitamin. So due to the deficiency of tryptophan, the body is not able to produce niacin vitamin, and the deficiency of niacin it's lead to the pellagra. So in Hartnup disease, the person will affected, resulting to the pellagra. So these are the references books from which this lecture is prepared.